title, Spark of Life, and it's a title that I stole from an article in a science magazine, and I'll get to that. So the Spark of Life here dwells on this Indian painting of a thunderstorm that's taking place, and in the middle of the squiggly line is a lightning strike, and we'll see the significance of that in a minute. So we've been blessed in recent times with exhibitions of Indian art in the United States, and that's where I've stolen these pictures off the web. And for the young people in the audience, it's perfectly okay to steal these images as long as you give attribution. And that's the most important thing in science. And these days it's very easy to grab things off the web and pretend they're your own. This is definitely not my own. So let me start very gently. It's a general audience here. So what is computation? So in the context of today's talk, I mean it to be the simulation of molecules in motion, in fact, typically a liquid, and probing this on a computer. And it's quite appropriate that I give this talk here because of Anisur Rahman was born in this city and has association with this city before he left and went elsewhere for his education. So I'm very, very pleased to be here. He was a dear friend, and I really do feel emotional attachment, even though I've not been here. So this loosely we refer to as computer simulation. And computer simulation is as old as computers. So this is an image of my grandson. He's now 12, and I think he was just 8 months there, and I was already teaching him. He's sitting on my lap, and I was teaching him, but in fact he's probably trying to make up his mind whether he's going to eat it or not. But anyway, I think you should begin young, and so he really didn't have much hope because his father's an electrical engineer and his mother was trained as a computer scientist, so the genes are certainly in the right place. Computer simulation of molecules, I said, we deal with mechanics and the origin of mechanics, and the way our field, and I'm not going to show equations, but our field really begins with Newton, and in the way we use it today, more about Hamilton than Newton. And of course, computing, we can't escape without mentioning Turing. We just passed the 100th anniversary of Turing's birth. And then the first people to use computers in the way that I'm going to use them are mentioned at the bottom, and they were physicists, all of them. Fermi, Metropolis, and Edward Teller, and then as we in the chemical physics interface use them, Anisur Rahman was the pioneer. But the first paper to mention computation is this one here, 1953. So we are already 60 years of this field. And you should take a look, I don't know if it's clear, but the authors of this paper, and we have Nicholas Metropolis, who is generally considered to be the inventor of this Monte Carlo, importance, the way to sample. You've got Ariana Rosenbluth, Marshall Rosenbluth, Augusta Teller, the wife of Edward Teller, and Edward Teller. So why were all these names on the paper? So we kind of speculate, maybe they were playing bridge or something, and they discussed a probabilistic way of doing things. But anyway, it's an unusual group. And as I've indicated here, this paper has 15,000 citations, so it's one of the great papers of all time. So computer simulation really belonged in the national labs of the United States. They were the only ones that could afford computers. And so that period I've put up there of about a decade, from the early 50s to the early 60s, this resided solely there. The computers were not proliferating around the world at that. Various countries were building computers, but they were not as good as the ones these people used. And these were the pioneers, and each of them had their own reason for using the computer to model something. I mean, we know these computers were also used to model weapons, nuclear weapons, but they were allowed to do science. But that was the heyday, in a sense, and I put this title there, When Women Were Computers. I might come back to that later. And how do we simulate on a computer something like water? So the first simulations of water were around 1970. So it took quite a long time to get from very simple models to the simplest liquid, in a sense, to get to water. And so the history of that depended on a number of contributions, but in particular a number of Nobel Prizes along the way, because if we're going to model these things, we have to understand how water molecules talk to each other, how do they interact with each other, and then it's unavoidable to skip the quantum mechanics of what's going on. So synthesizing and integrating these various tools all come together, and we generally acknowledge, those of us in the field generally acknowledge Anisour Rahman as the father of it from our perspective. Nowadays, we're limited mostly by our imagination about what one could or could not model. And I'm just showing a clip of some work done by a student in physics at the University of Pennsylvania. He came over to my group and said that he was building a chemical nose in the lab. It was a carbon nanotube that had strands of DNA on it, and in the lab he could make this. It was a very difficult experiment because the nanotube is, of course, nano in size, so you don't see it with your eyes. You have to image this thing, and then you want to put some DNA on it, and then you want to make it part of a device, and then the idea was that if particular chemicals passed over this device, it would respond in an appropriate way. And what Bob Johnson and his thesis advisor, Charlie Johnson, they both called Johnson, had discovered is that this thing was a pretty sensitive detector of explosives, so it wasn't surprising the Army Research Lab was also interested. But he came across to my group and said, could we model that? And so this is basically a big box of water. The water molecules are not shown. It's a particular strand of DNA, a particular sequence of bases, and this thing can be done using a big-ish supercomputer. This was already done a few years ago and used a couple of thousand computer processors running totally dedicated for 1 month, and then you produce this movie. Another extreme of example from my group from the same era was a cover on Science magazine, was trying to simulate a vesicle made out of lipid molecules, and how does this thing form? And the size of this vesicle is tens of nanometers across, and it's actually the vesicle that is used in the signaling in your brain that delivers the chemical sensor to acetylcholine receptor. A motor protein carries this vesicle on its back along a filament, walks, and then drops this at the synapse, and that's the start of a signal. So again, this was state-of-the-art. Collaborator Wataru Shinoda that spent a couple of years in my group in Philadelphia, and we modeled, and on the cover of the magazine is trying to model what would happen when this synaptic vesicle docks at the plasma membrane. So these sorts of simulations, as I said, we're limited mostly by our imagination. But today's story begins with Galvani and this lightning strike, and for those of you that know that Galvani was dissecting a frog's leg during a thunderstorm, and the frog's leg reportedly twitched. And it's that twitch that's going to be the focus of my talk. So if we fast forward 100 years from Galvani, and there was a Nobel Prize given to these two gentlemen here, Golgi and Carnell, for anatomy. It was the prize for physiology or medicine, and they had, through some very clever staining techniques under microscopy and anatomy able to reveal the structure of neural net, the neurons in your brain, basically. And these images that I've just copied here, up here, these original images of Carnell from staining are still used in textbooks to this day. So in fact, those original images inspired the painting in the lobby of a new lab in Florida devoted to neuroscience. So that's a way of background. So in a sense, the story began 200 years ago or so and was already honored 100 years ago by the Nobel Prize. So this is the article that got my attention. Peering into the Spark of Life was a commentary on a paper which reported the structure of an ion channel. And the ion channel in question is up in the top right. I'll say more about it in detail. And this structure was the channel that allows sodium ions to pass into a cell, what's called an excitable cell. It's a bacterial version of it. It's not actually the channel that we have in our cells, but nevertheless it's a functional channel, and its structure appeared in Nature 2011. And those of us that do computer modeling, just as those of us that are interested in solid state, it's often helpful to know the structure of the system you're interested in. So this was a real breakthrough for the field to get some insights into the structure of this channel. And I'll say a bit more about why this is useful. So this is a straight quote from that commentary on the structure paper. They play a crucial role in triggering the electrical event. So if I decide I want to move my arm, the muscles have to be told to do something. And the fact that it takes much less than a second for this to happen is something that at the back of my mind leads me to believe we have a hope of modeling the fundamental events on a computer, because we are limited to the time scale we can model. So the crystal structure reveals how this channel might work. And the key is here that the sodium ions have a crucial role to play in what was called the action potential. And I'm going to say a few words about the action potential. So let me do that. So looking at the paragraph that I've copied out here, when a frog feels like jumping, the idea begins with a spark, a bioelectrical event. And this is a trace of action potential. I just took that from Wikipedia. This is the typical potential that exists across an excitable membrane. And when the voltage changes, the sodium channel, which is the one whose structure was just published 3 years ago, it opens and sodium ions rush inside the cell. If that was to continue, that wouldn't be very good, because there will be an accumulation of charge. And as we know, the charges are not happy if they keep pushing positive charge in. So what happens then is a potassium channel opens, and that reduces the potential, and this whole thing recovers in a few milliseconds. So the whole process takes place reasonably quickly. I'll say a bit more about that to help clarify. So the basic structure of this bacterial analog, the structure was determined by a Carnell group. Carnell is professor at Washington University in Seattle, and has spent his whole lifetime studying sodium and calcium channels. This character here, Nin Zheng, is the crystallographer that did the crystal structure work. So the collaboration between this pharmacologist, physiologist, Carnell, and crystallographer produced this beautiful structure. The bacterial version of the channel is an oligomer, a combination of 4 identical proteins. And they are color-coded here in the 4 colors you see: white, red, green, and blue. And each of these proteins consists of 6 alpha helices. And when they come together, you have 24 of these helices. And the outlying pieces are 4 by 4, so 16 alpha helices arranged 4 by 4. And these are the so-called voltage sensors. So I should say that this channel is floating in the cell wall, in the membrane, the excitable membrane, the coating of the cell. And these 4 outliers are the ones that detect the electrical signal. And the middle pore is where the ion is going through. And the asterisk there marks the pore. Again, let's go back to this basic structure. The asterisk marks the pore, where the ions go through. And these 4 outliers are the sensing domains, as they're called, the voltage sensors. So let me go back a bit to the story I was telling you about the action potential. So here's a cartoon of a cell, and in the middle of a cell, the concentration of potassium ions is 150 millimolar. The concentration outside the cell is about 150 millimolar in sodium. So we know our bodily fluids contain a lot of sodium, and that's why they tell you to watch the salt you're taking. But nature's been very clever. It doesn't use salt inside the cell. It's using potassium, which is the minority constituent. So, as I said, when you want to start this electrical signal, the channel that I was just showing you opens, and sodium rushes from the outside inside the cell, and there will be an explosion unless the potassium, that's the only thing in excess in there, comes out. And that's what re-equilibrates the potential. That is the basic events that have to take place. So actually we have to understand not only sodium channels, but also potassium channels. We know a lot about potassium channels, and I'm going to review a bit of that history for you. But I've got ahead of myself, and I'm going to switch the clock back to 1939, when a paper came out in Nature again by 2 young scientists. And there's a very interesting story here. The thesis advisor was age 25, and the student was age 21. And I would like the young people to read this abstract, because we're in an age where everybody makes breakthroughs. Everybody is the first to do something. Everybody's done the biggest and the best. Just read this abstract from Huxley and Hodgkin. So Hodgkin was the senior author, Huxley was the junior one. So nervous messages are associated with electrical change known as the action potential. I went through that. This potential is generally believed to arise at the membrane around the cell, which is situated between the external medium and the interior one. If this theory is correct, it should be possible to record the action potential between an electrode inserted in a nerve fiber and the conducting fluid outside. And then most nerve fibers are too small for this to be done, and we recently succeeded in inserting a microelectrode into the giant axon of a squid. So when you look in detail at the paper, and these were 2 gentlemen in Cambridge, they went to Plymouth so they could be near the sea, the ocean, and get the squids to study. I really want, these guys shared a Nobel Prize for doing this. So they didn't tell everybody it was the first. They did a lot of other things too. So that was 1939, and then the next paper in their series appeared in 1945. And they mentioned that the bulk of the work was done in 1939, but the World War II interceded, and these guys were doing other things at that time. And then they came back to their original work. But what happened was that Huxley built an analog model of the processes I just talked about, which is still the basic model we use today. That what we needed was the membrane was essentially a capacitor, the sodium current can go in one direction, potassium can go the other. To some extent, they're reversible as well. There was also a leak current, and I don't want to get into that technicality here. It's not so relevant. So they built a model of the excitable membrane, even though we didn't really have the first papers on the structure of biological membranes till the 1970s. So this predates. I mean, there was a lot of supposition here. In the end, they were able to deduce, Hodgkin was the senior guy, and he did experiments, but he was able to deduce that potassium ions, remember I said sodium is the first event, it rushes in, potassium comes out to equilibrate. They were able, by some measurements I'm going to show you, to deduce that potassium ions moved in single file. And I'll come back to that. So what Hodgkin did with Keynes was to build this kind of analog computer where they did an experiment where they made like a baking tray with ball bearings in it, and they joined the two together with a tube, and they shake this thing, and they, by coloring the two, the steel balls on each side different color, they can see whether they go backwards and forwards. And somehow from this, they were able to deduce the properties of the potassium channel. They were honored with a Nobel Prize in the early 60s, and unfortunately neither of them are with us now. This is a photograph. The last time I saw Huxley, he looked like that. I mean, that was pretty good copy of him. I think he was 92 years old at this point. And I was at a dinner at Trinity College, Cambridge, and before dinner people have a glass of champagne. And he was so eager to get his glass of champagne, even with his walking stick, he managed to push me aside. And so he was a master of Trinity College and Nobel laureate. And Hodgkin is up there. So these guys shared the prize with another Englishman called Eccles. Now, that's a bit of history, but remember what I said, that inside a cell is potassium, outside the cell is sodium. Sodium rushes in, potassium comes out. That can't be good because the cell is designed to work with potassium. So some clever character intuited that there must be another of nature's machines that puts it right, that now switches the sodium and potassium back together. And this gentleman here, the Danish scientist, already speculated about this protein and eventually was honored in 1997 with a Nobel Prize for this machine that is able to exchange potassium for sodium and re-equilibrate the cell. In the meantime, there was also another Nobel Prize in this field for being able to record single ion passage. Patch clamp it's called. It's the standard workhorse in the field. And there's another Nobel Prize. So you get the impression that this is a nice area to work in. It seems to collect pieces of gold quite frequently. So the next event in the area in potassium channels was 1998. The cover of Science magazine reported the structure of the pore of another bacterial channel, KCSA. It's unusual in the sense that it didn't have the voltage sensors. This particular channel is just the pore. And so McKinnon is the guy that crystallized this, did its structure at a synchrotron, actually Cornell University synchrotron, and published this paper in Science magazine 1998. So we already had a knowledge of the potassium channel, and these images are taken from that Science paper. And in particular, why it caused so much excitement was that they were able to, in the crystal structure, to see the potassium ions and where they were. And the remarkable fact was the prediction of Hodgkin and Huxley that these ions would pass through in a single file one by one is revealed in the McKinnon structure. Actually, McKinnon did something very simple. By using the fact that sodium, potassium and rubidium are almost essentially the same size, he prepared a crystal with potassium bromide solution, and then he replaced that by rubidium bromide and was therefore able to phase his structure and get the position of the ions, which is the remarkable thing. That led to some interesting comments and put in place a lot of the ideas which weren't quantitative but were qualitative about the action potential. The idea goes as follows. If you want to move your arm, we want the sodium ions to be able to rush in very fast, so you would intuit that the pore of the sodium channel is likely to be much larger than the pore of a potassium channel. So basically, if you want that to actually open, you want it to fire very fast. Another thing with the sodium is that sodium ions are very tiny and they grab hold of water molecules very hard, and they're not very likely to let go of them. So again, you might intuit that the pore of a sodium channel would have to be big enough to take the ion with its full hydration sheath to pass through. And so there was a commentary by Clay Armstrong going on with this paper saying that this structure helps us rationalize the known characteristics of the action potential. So I already mentioned that McKinnon got a Nobel Prize literally 5 years after the science paper. Rather remarkable. So it was the McKinnon paper that triggered the people in my community to start modeling these things. I mean, we had the first structure. And what we typically use are these big computers, which are now available around most of the world, so there's no special privilege anymore to being in the United States. Certainly Europeans have big computers, and even here you have a very decent facility by any standard. In fact, much better than the one I have at my university in the U.S. So again, you're fortunately here limited by your imagination. So that's the machine that we used in my group to study these things. So I don't want to leave you with the impression that my group is the only group interested in these ion channels. I've listed a bunch of names of people up there. Some of the people are in the US, others are in Europe and elsewhere. And it's a very active community. That's the main take-home message. Just to say something about the difference between eukaryotic, that is, mammalian cells. So up the top, I show you top panel A gives you in a cartoon form the KV, the potassium voltage channel, 6 alpha helices. You can see 4 of them labeled blue, 2 of them are red, and there's a tiny little piece which is also there, which is the so-called selectivity filter, which ensures that the potassium pore will only let through potassium ions. And the bacterial structure is an oligomer of 4 of identical versions. But in the sodium channel, the eukaryotic sodium channel, you have 4 different voltage sensors. The voltage sensors are not the same on each of these. So the human versions of these channels is much, much more complicated than the one we are modeling. So McKinnon's structure was only the pore. There was no information on the voltage sensor. We know its sequence, of course, and people have tried to guess what the structure is. So I got involved in a collaboration with postdocs and other collaborators around the world to try and understand in much more detail how does the voltage sensor work. In other words, you have a channel. It can't be open all the time. It has to open in response to an electrical signal. I want to move my arm. So a lot of the time it's going to be closed or resting. So what are the motions that take place in this assembly of proteins that cause the opening and closing of the pore and sense it? So that was work that the first clue came, yet another structure from McKinnon in 2005, where he was able to look at this so-called KV 1.2, this one here, which is a voltage sensors, a system with a voltage sensor on it. So what we did with the collaborators I have here, this is in fact a three-way collaboration between my group and former postdocs in France and Brazil, again showing you that the world is flat as far as science is concerned. We can work with anybody we want due to the web. And what we were able to do was start from the McKinnon structure and try to understand what are the processes that take place when the voltage sensors drive the channel to open and close. So this was a paper that came out in Proceedings of the National Academy of Sciences. I'm not going to spend time on the detail. The most important finding is that the sensing machine is composed of oppositely charged amino acid residues, positive and negatively charged, and to open and close they have to click past each other, and it's a very complicated process, but eventually you have to drive all the way down. There are at least five states of the voltage sensor on its way from open to close, and this paper goes into the details of that with one of the first insights. I don't have time to go into the gory detail, and I don't think it's relevant, but at least we were able to make a structure that was open and closed. So that was the state of the field 3 years ago when the paper came out on the sodium channel. So, and I already mentioned this paper of Carnell. Why should you care about sodium channels? So I've got here some quotes from Bill Carnell, who is the guru of this field. And just to calibrate you, his Hirsch index is 124. So just in the field of great scientists, he really is a great scientist. But just read the one at the bottom. So if you go to the dentist and you don't want to feel pain when they're drilling in your tooth, you get a shot which actually closes the sodium channel so that you don't feel pain. In other words, the signal's not getting to your brain that something nasty is happening. So this is why we are very interested in understanding the function of these channels, and in fact the misfunctioning of such channels which are related to diseases. And it's a very active area in my group, and I don't have time in this lecture to go into that, but that's what drives a lot of our interest. So just to summarize, there are 9 or 10 of these different sodium channels that have slightly different functions in us as human beings, and if there's something wrong, if there's a mutation wrong in the voltage sensors of these channels, you can suffer from these diseases which are called channelopathies. And we're very interested, particularly these here. As I said, unfortunately I won't get into that detail in this lecture. Anyway, what did Carnell find? This is the basic structure that was published in Nature 3 years ago. That was the structure, and indeed the pore turned out to be very wide, as we would have intuited, and it was reassuring to see that. And this is an overlay of the potassium structure from McKinnon and the sodium pore from Carnell's work, much wider. So let me keep moving here. So once this structure came out, my collaborators and I worked extremely hard to try and find out some of the gory details about how this would work. So here's a snapshot of a typical system we would study on the big supercomputer. It would contain the whole assembly of 4 of these membrane proteins, the water, the sodium ions and the chloride ions to make it neutral. And those are the things that we do. And typically we do a very long run. You can see faintly here, this membrane protein is wiggling around. It's embedded in a membrane, and there's an ionic solution there. And we have to run millions and millions of time steps to watch the protein evolve. And what we were interested in first was identifying the binding sites for the sodium in the channel. And I won't run this on and on. There's no need, because the upper image shows there are two primary binding sites for sodium that we find in this simulation, which came out a few months after the crystal structure. So I'm going to stop that. So there are two sites. But one of the things that was fascinating to us is the coordination structure of the sodium ion, which might be octahedral or square pyramid. But we noticed a very, very surprising phenomenon. That with the ions in the channel, in their pockets, so it's not conducting at the moment, the ions are there passively, water molecules could come from outside, go through the channel, go all the way through the pore, come all the way back and go back and visit the bulk solution. So obviously it's not going to be harmful for water to do that, but this channel is sufficiently wide that water can permeate through. Another thing we observed was that water could permeate through these voltage sensors, and that's not such a surprise if you think about it. The voltage sensors are composed of positive and negatively charged amino acids, and they would like to be hydrated, their charges, so that's not a surprise. But you can imagine now, if there was a defect in that channel, you might get a small probability of ions to go through the voltage sensor. And this in fact happens, and it's called an omega current by the physiologists, and is a very important measure of whether your channels are behaving properly. Well, once we had a working structure, and once we could do these long simulations, somewhat routinely, fortunately we were given liberal access to a computer, you can play games like adding another sodium ion and asking, how does the conduction work? So this is a case where this particular structure we're looking at is actually a closed channel. The bottom is closed, and there's enough room for about 50 water molecules in this cavity underneath the filter region. And in this case, we put an extra ion in there, and we get a kind of crystallization of these ions, but we didn't see conduction. So if you add another ion, which is effectively putting a large field across, or electrical potential across the channel, eventually we'll see here at least one conduction event. And so what you have to do, it's a bit like particle physics, you have to do lots of trajectories, and you try to get what is statistically meaningful. But you will actually see a conduction event here if we let it run long enough. There you go. So in fact, doing lots of studies like that, we can get to understand the conduction mechanism. But in fact, this structure that was published by Carnell was a closed channel. We didn't learn anything. Actually, conduction has to happen with an open channel. So in my group, we spent a lot of time trying to make on the computer an open structure. And this is a simulation we carried out by applying very, very gentle forces to the bottom of this pore region to open it very slowly. And eventually it opens, and we get an open structure, which will enable us, and did enable us, to study conduction in an open channel, which is what makes sense. Should I let that run on a little bit longer? You'll see little red dots, which are water molecules, eventually coming through an open channel. You have to do this very slowly, otherwise you will damage the protein. You will cause some amino acids to go into the wrong conformers. I think at this point it's more or less open, and water molecules can go through. It turned out that some very clever experiments were carried out about the same time as we were doing the simulations, and in the panel labeled C there's an overlay of the structure we made on the computer with one that was measured by NMR studies. And in fact, the overlap is very, very good. I'm not going to say any more, but now let me go to this conduction in an open pore. So we were able to make an open pore, and therefore we can actually study the real conduction mechanism, so to speak. The pore region of the filter region that in the potassium channel is very tight and narrow, in this sodium channel you can see that the conduction occurs. And in the write-up, the scientific paper, we talk about the role of this aromatic residue at the bottom in steering the ions through a cation-pi interaction. But in any case, we can study this in gory detail. So if anybody's interested, this paper that came out again with this US, Brazil, France collaboration. I want to just say a tiny bit about selectivity. In fact, because the bodily fluids have just a minority of potassium, the sodium channel doesn't have to be particularly selective. What you really want is to get the electrical charge going. And so there's very little potassium outside. So it's not essential for the sodium channel to be selective. But the potassium channel has to be selective. So, and we understand that. And I, again, don't have time to go into all the detail, but this is one of the things we've studied. I just want to say that this is an incredibly active area, and this kind of work is usually reported at the biophysics, the annual meetings of the Biophysical Society. And at the last one, there were at least half a dozen groups around the world, from Australia, from Germany, from England, and so on, all having an interest in this mechanism of conduction and the sodium channel. It's a highly active area. Since that first paper by Carnell, I'm just going to flick through a few more papers by other groups that are doing related things. But let me just summarize by saying that the computation has played a key role in identifying these binding sites and discussing the conduction mechanism, and indeed even contributing to understand the open structure. So I'm going to move on to talk about something else which is very dear to my heart, which is the interaction of these ion channels with small molecules. Now, I don't know if anybody here is familiar with the Japanese fish fugu, or the blowfish, which is deadly poison. And the deadly poison is a toxin which blocks the channel. And that's something we're interested in my group, but I'm not going to talk about that. I'm going to talk about something slightly different. And the great chefs in Japan just leave a little bit of this toxin that makes your gums tingle when you eat it, but shouldn't kill you. So that's apparently a skill. So we work on model systems, and so do the physiologists in the field. I already alluded to the fact that the sodium channels in our body are very complicated, and Carnell some years ago was making a pitch to use this thing called Natchback, which is a stripped down version, a small version of this particular channel, and it's a tetramer, it's an oligomer, and it forms an oligomer. And so one of the projects, and it's the last thing I'm going to talk about that we've been working on in my group, is trying to understand how anesthetics interact with these ion channels. Let me just take a minute to talk about anesthetics. When you go to have surgery and you're treated with an anesthetic, I already alluded to the dentist giving you a shot. Anesthetics are typically smallish molecules, and an anesthetic has to have, and there are lots of small molecules, an anesthetic has to have at least three important properties. First of all, it has to put you to sleep, okay? So that's the most important thing. But you should wake up. There are lots of small molecules that put you to sleep and you don't wake up. So And many of them cause accidents all the time, like carbon monoxide. So, as I said, putting you to sleep is important, but you better wake up. The next thing that's very important about an anesthetic, so it has to be a small molecule, because generally, not always, given gaseously through a mask. The next important thing is you should have amnesia, because the surgeon is going to cut you open, and you don't want to remember this pain. So you go sleep, wake up, you want amnesia. And then the third most important property is it should be an immobilizer. The surgeon doesn't want you wriggling around on the table. So there are subsets of small molecules that do 2 out of 3, but that's no good. So actually the subset of molecules that can be used as anesthetics is very small. And for the past couple of decades, my group has been collaborating with anesthesiologists, trying to understand how these things behave. They are typically the most dangerous drug that you get exposed to as a human being. And there are millions and millions of surgeries performed every day by empirical rules. And so this is an area that I've been working in for many years. So in this particular context, I should say that one of the guiding principles for anesthetic type molecules comes from more than 100 years ago, where a couple of gentlemen, Meyer and Overton, suggested that solubility of a gas in olive oil is a good judge of whether this will work as an anesthetic. And this correlation is used even today. So my early work in my group was concerned with partitioning of small molecules anesthetics into the cell membranes, but we've extended that now to look at the interaction with membrane proteins. And the motive for doing this was a paper by a very eminent, very eminent anesthesiologist, Hemmings, and a paper he published just a few years ago saying that this particular model, this stripped down version of a sodium channel that Catterall had proposed, in his lab at least, with work with Anna Correa, suggested that this would be a good model. So I'm just going to show a computer simulation of what we call flooding. We have a system of a membrane, a lipid membrane, embedded in it this sodium channel, and then we put into the solution, in this particular case, 80 anesthetic molecules, isoflurane. We've studied other molecules too, and we watch the partitioning from the bulk solution into the membrane, and then from the membrane into the membrane protein. And this is a very long simulation, but in the end we can identify the binding sites of these anesthetics. Now we know that the binding sites must be weakly bound. Why do we know that? We know from experiments that if you're under anesthesia and they put you in a pressure tank with a few atmospheres of pressure, you actually wake up instantly, a few seconds. So it's reversible by just a few atmospheres of pressure. So simulations of this type allowed us to identify binding sites and quantify them. But one interesting thing of the sodium channel is that the mechanism of partitioning from the membrane, and I've got another little movie that shows that. In fact, the anesthetics come from solution from the outside, they dissolve in the membrane, so this idea of the Meyer-Overton rule of partitioning into lipid is very relevant. But then they come in between the proteins, these alpha helices, there are gaps, and these anesthetic molecules here, they dive into the middle, into the pore region, and they block the pore. And so that we think is one of the primary binding sites. There are other binding sites. I'm not going to run that movie, I'm going to move on. There are other binding sites at the top of the pore, and you can see the fourfold symmetry here, and those are sites which we think the toxin binds. Remember I mentioned the toxin, which if it blocks that channel, you can begin to understand why it might kill you. And there's yet another binding site that we identify, which is a crucially important one, and it's here. And that is the linker between the voltage sensor and the bottom of the pore. And so when the voltage sensor tells the channel it has to open or close, if there's anesthetic there, it will inhibit the opening and closing of the pore. So this work's been published in this journal here, Computational Biology, and that's a general area that we're very active in my group, studying how drug molecules would interact with these kind of channels. So I think I'm going to stop there, and I've got one more image I'm going to show you, is our most recent paper came out in the Journal of General Physiology, where we talk about the design of these nature's machines, these voltage sensors, which are crucial. And as I say, the voltage sensors, if something goes wrong with the voltage sensor, there are many nasty diseases that you're going to inherit because of your parents, because of genetic reasons. And it would be very nice to be able to mitigate these unfortunate channelopathies. And so that's an area that's of great interest to my group right now. So I'm going to close there. Thank you.